Diversity of Life, Investigation 3.2a, Paramecium. Hi everybody, it's Mr. Kennard again. Let's review a little bit from our last lesson. We talked about a plant called Elodea. It was an aquatic plant that people have in their aquariums sometimes and things like that. We used a microscope in this case to look at some of the smaller microscopic structures that make up that plant. And what we found was they had bricks and brick-like structures that had things in them that helped the cell, were labeled as a cell, those brick-like structures, and then found that there were structures inside of them that actually helped the cell do the living, the purpose of living. We talked about cell wall, cytoplasms, chloroplasts. We looked at some other things, mitochondria, nucleus, all of those. When we had our wet slide, did we notice, was there anything else moving? And we're going to focus on what potentially could have been some of those moving things, something called a paramecium. That's the focus of this lesson. Before we look at our paramecium, we're going to spend a little time to look at that evidence of life chart. Question 1, what was your evidence that the Elodea plant is a living organism? Go through that checklist and see if there's anything that you saw from the Elodea if it was going to be living organism. 2 was, did you see anything else in the Elodea slide that you might think might be living? Many of you, when we talked about things, what makes something living is whether or not it can move. So what you saw, if you were able to see things moving around that Elodea plant, a lot of them might be a water-based kind of object, a living thing, potentially a moving item called a paramecium. And we're going to focus on those paramecium that we may have seen in that Elodea water wet slide. Now that we've seen what Elodea are as a plant, remember they were plants and all of the cell structures that went in it, what is it? A paramecium. And we're going to focus on how they are alike and how they are different from each other. Many things might, if they're living, they might have similar things that work together or work in concert with each other, or might have other similarities that because they're both living, they share. And they might be some different things because a paramecium is probably not a plant, just like humans are not plants. So we're going to focus on, that's our focus question, how are Alodia or Elodea and paramecium alike and how are they different? All right, at this point I need you to download or have available in a split screen that Paramecium observations worksheet. We're going to fill that out as we see and try and find some paramecium. This little video is going to show us about how to set up a wet mount slide for paramecium, both before and then after a slowing medium is added. They're going to add something to slow them down because they're a little fast, little boogers. So before we do that, you're going to want to describe the movement of the paramecium and behavior as well. So let me show you that video and we will go from there on how to set up this wet mount slide.
Put one small drop of paramecium culture on the center of your slide. Do not put a cover slip on. Put the slide on the microscope and don't forget to turn on the light. Focus the microscope at 40 power to make sure you have paramecia on the slide. When you have found paramecia, increase the magnification to 100 power. Then describe the movement and behavior of the paramecium.
At this point, that's what we're going to be doing for in-class students. We're going to want to find them, we're going to want to isolate them, and we're going to want to describe their behavior on that observation sheet. Once that's done, we will see if we can slow them down a little bit as they are fast movers. One way to do that is with a medium. We'll add this drop of this slowing agent and it will give you a little more detail in the video.
Remove the slide from the stage and add 1 drop of methyl cellulose. This time, put on a cover slip, and if necessary, blot up any extra liquid. Put the slide on the stage and search for one paramecium that is still moving. Focus under low power, and then increase to medium and high power. Focus using the fine focus knob. Describe the paramecium and draw it in the circle on your notebook sheet.
For those of you in class, you were hopefully able to find a paramecium and to draw and describe its movement, or even as it slowed down. For you, those of you that are virtual or missed it for whatever reason, or if you weren't able to find one, what I need you to do is to go to the online resource again, the fossweb.com. Let me move that back up here, hang on. fossweb.com. It's terrible writing. And find the database section of the multimedia. In there you will find this Paramecia collection. And those, I'm going to show you some of the videos that are in there in case you don't want to visit there. But if you do, you can go back and watch this video again, or find it in the database and find some of the Paramecium and how they move, sizes, etc., that you can then answer and fill in the information on your sheet. Here's an example of a paramecium that's been still photoed. You can see its shape, its structure. It looks kind of like a cigar, they like to call it. You can see some of the smaller pieces inside, and if you look really closely around the outside edge, it looks like it's got a little bit of hair. This is actually part of about an 8-second video of movement, and let me show you that now. So yeah, as you can see, they kind of spin and twist and move within the water, within the liquid that they're in. Yeah, they're able to move completely on their own. That is a paramecium. I've got a couple more to show you as well too. Here's another video with some more movement and interaction with the paramecium. Pretty cool. Notice how they were spinning around, and if you look closely, you can see there's a lot of other cellular structures inside of those cells, much like maybe what you saw within the Elodea cells. Got a couple more little videos to show you. It should be really cool to look at these paramecium. This is a video on paramecium that I'm going to actually save to the next lesson because it covers a lot more stuff that we're going to cover in the next lesson. So we'll move past this. So let's now debrief our observations about the paramecium. What were some of the movements that you might have seen with the paramecium? Well, I know I saw that they're moving through the environment, they're bumping into things, they're turning around, maybe moving circles inside the paramecium. A couple of them bumped into each other. Some of them move at different speeds than the other ones as well. So those are the kinds of things that you're going to see from paramecium in their water. Number 2. What did you see inside of the paramecium? Saw a bunch of things, blobs and bubbles and dark areas and shadings and all sorts of stuff, circles, all different kinds of structures maybe be inside there as well. What did you maybe see on the outside of the paramecium? Maybe a thing like there were some folds, some grooves, maybe skin, maybe little hairs. All those kinds of things were what I saw on the outside of it. And then approximately how big, what was the length of the paramecium? It would almost fit all the way across the 400x. So if that was the case, almost 400, almost across would have been, all the way across would be 0.4. So maybe in the neighborhood of 0.3 millimeters would be the length. If we got it to hold still, that'd be the hard thing. So yeah, lots of different features about that paramecium that would be different than the Elodea. Finally, our last question on the observations is, how did the size of the paramecium compare to the size of the Elodea cell? In this case, the paramecium was much bigger in this case, a lot bigger. Paramecium, we said was about 0.3 millimeters. And the Elodea was a lot smaller. One of its cells only worked out to about 0.1 millimeters or so. So there was a difference in terms of the size. Let's now go back to our evidence of life chart. Are Paramecium living or non-living? And what would be your evidence for that? We could probably say that they're moving. We could probably say that some of those things inside of it maybe acted like the Alodia did, or the Elodea. And then, could you see if it was doing all the things that were listed by the definition of an organism? We went through those 8 characteristics of life. Were we able to see if it was doing all of those things? That becomes the big question mark. Maybe we need more observations and more evidence to make that determination. It sure looks like it, though. Did you see it eat? Did we see it use energy? Did we see it give off waste? Did we see it reproduce? What might we do to see some of these activities? I think we would have to do some more investigations and do some more observations of a Paramecium as it goes through its life cycle, or just additional viewings. And we're going to look at that in the next lesson. Thanks, everybody.